and it's a pleasure for me to join you all this morning. I think it's still morning for you, and talk about one of my favorite subjects, the immune effects of oligonucleotides. Just a disclosure, I do work at Checkmate Pharmaceuticals, where we're actually developing immune stimulatory oligonucleotides for cancer immunotherapy. They have great potential as drugs to activate immunity, but what we want to talk about today is the downside of that. And for people trying to develop ASOs or siRNAs for other applications, you really don't want the immune stimulation. And so the key points I'd like to touch on in the next 15 minutes or so are, first, why did the immune system evolve to be activated by nucleic acids? Because this is most definitely not an accident of biology. And to make several points that Patrick also made, one is that the immune responses to nucleic acids often are species specific. And so if you're using mice to screen for humans, they're really not perfectly predictive. In addition, the formulations and the nucleic acid modification can greatly modulate immune stimulation. I'll talk a little bit about that, but there's a lot more to it, and Patrick made those points as well. And then I'll try to dive into, just from a very practical perspective, what are some of the screening assays that are best at picking up the immune stimulatory effects of nucleic acids. So first of all, in terms of why we have this, it actually, I think, is fascinating from a biological perspective, that really what this is all about is protecting genome integrity. And from the earliest organisms, bacteria and other organisms, if you can't protect the integrity of your genome, then you don't exist as a species, because your genome gets taken over. And so all kinds of mechanisms have arisen through evolution in order to protect the genome. Nucleases, restriction modification systems, CRISPR, and of course we take advantage of these therapeutically. So RNAi obviously evolved as a defense mechanism against foreign nucleic acids. We can use that therapeutically. And in their most highly evolved form, the receptors in mammals, the toll-like receptors and others, are very sophisticated at being able to detect foreign nucleic acids. Now, along the way, the detection of nucleic acids also will occur with self-nucleic acids to a certain degree. And stress of our own cells can release our own nucleic acids. And sometimes those are structurally the same as foreign nucleic acids, and sometimes they're different. So I do want to add that it is a little more complex than just detecting foreign nucleic acids. So let me dive now into some of the specific receptors and the pathways that we need to think about here. And I'll start first with RNA. The point being that foreign RNA is typically initially taken up by cells and encountered in the endosomes. Whether it's from a virus or a lipid nanoparticle, the initial interaction with the cell membrane leads to internalization into endosomes. Cells generally do not have membrane receptors that are highly activated by nucleic acids. They do have membrane receptors that can bind nucleic acids. So inside the endosome, you have the first line of detection. And for RNA, 3 receptors are important: TLR3, TLR7, TLR8. Now, as everyone knows from the antisense and siRNA fields, nucleic acids tend to get concentrated in the endosomes. And the rate limiting step for efficacy is release into the cytoplasm. And so one of the implications for that is that the concentration of therapeutic nucleic acids is very high in the endosomes, and it's relatively low in the cytoplasm. So it's much easier for the immune system to be activated in the endosomes than by cytosolic receptors. RIG-I and MDA5 are 2 of the major cytoplasmic receptors that we think about for detecting RNA. And conversely, you're more likely with the therapeutic RNA to bind to receptors like this than you are to bind to whatever your mRNA target is inside the cell. So we have nucleic acids, they come into the endosomes, they may be interacting with these TLRs, and there are several downstream pathways that can be engaged to a different degree depending on the sequence and the modifications of the nucleic acid. One of these pathways is the IRF pathways, IRF3, IRF7, these are interferon response factors, and they, if activated, will lead to induction of type 1 interferons. The second major pathway from this set of TLRs is the NF-kappa B CPG pathway, leading to production of pro-inflammatory cytokines like IL-1 beta, TNF, IL-6. And so if either or both of these pathways are activated, you'll have production of those factors, and you can measure that. And then with the cytoplasmic receptors that can be activated, again, it's very similar downstream pathways. Again, you have IRFs that can be activated, leading to production of interferons. And in addition, there is another pathway, the inflammasome pathway, that can be activated by some nucleic acids. And that again leads to production of IL-1 beta. So one point just to mention is that by screening for those factors, you can detect if any of those pathways are being activated by nucleic acids. You don't necessarily know exactly which pathway is being triggered, but if you're just trying to develop an ASO or an siRNA that isn't activating these pathways, that's one way to go about it. Also, the reporter cell lines don't necessarily measure all of this because they are typically using an NF-kappa B reporter, and some nucleic acids don't really activate NF-kappa B as much as they activate the IRF pathways. And so I think it is important to use an assay like a PBMC assay to detect that using human relevant cells. So a few points to consider here is that, again, as I mentioned, some of these pathways are cytoplasmic, some are endosomal. It's generally harder to achieve endosomal escape and to activate the cytoplasmic pathways. And of course, just because you may design an RNA that you intend to activate RIG-I, and one of the distinguishing features of those is a 5' triphosphate and a blunt end, the RNA inside that could potentially also activate the other TLRs. And so if you're trying to determine the mechanism, you need to include a 5' monophosphate control that cannot activate RIG-I, but could still activate the TLRs. In mice, TLR8 isn't functional, and TLR7 may substitute for it. But if you're developing something in humans, it's just a good idea, again, to be aware that mouse data don't predict this. And because of the molecular structure of TLR7, different RNA and TLR8, different RNA sequences differentially activate those two TLRs. And that's important because different cell types express TLR7 or TLR8. And so some RNAs may activate TLR7 expressing cells, macrophages, for example, but TLR8 expressing cells, like many monocyte populations, might not be activated by the same RNAs. Human TLR7 does have two binding sites, and RNA can activate both of those binding sites. Now, there are small molecule ligands for TLR7/8 that are often used as positive controls. R848 is an example of that. The small molecule ligands really only bind one site. They don't bind both sites. And so they actually have a very different cytokine effect compared to RNA ligands. They induce much less of the type 1 interferons compared to an RNA ligand. This just shows the molecular structure of the TLR7, and TLR8 is very similar, where you can have RNA sequences, especially poly-Us, binding to the TLR7 at both of the two sites, but R848 just binding to the one site. So DNA sensing pathways, now turning to those, pretty much the same paradigms here. Again, internalization of DNA into the endosomes initially. There's just one TLR in the endosomes for DNA, that's TLR9. And that is activated by CpG DNA, the CpG standing for cytosine phosphate guanine. And the idea being that in our own genome, when a C is followed by a G, the C is usually methylated. And what TLR9 is activated by predominantly is unmethylated CpG DNA. And once that is activated, there are again the same downstream pathways, IRF7 leading to interferon, type 1 interferon production, and the NF-kappa B pathway. And just as with RNA molecules, different TLR9 ligands can differentially activate those two pathways. So you can get different amounts of type 1 interferons and inflammatory cytokines like IL-1 beta, TNF, IL-6, depending on the backbone and modifications of the DNA. And then in the cytoplasm, we have STING as the major cytoplasmic receptor for oligonucleotides. Not so much of an issue because you don't normally accidentally activate that, but that pathway also can be activated by endogenous DNA released from the nucleus into the cytoplasm, and it may be important in homeostatic mechanisms. One point about the CpG DNA for TLR9 TLR9 is that when TLR9 is activated, there is an obligatory step where DNase 2 needs to cleave the DNA in order to get the full activation of IRF7. And using phosphorothioate or other modified DNAs, that step can't happen, and so you don't get that pathway being activated. In terms of species-specific differences, mice express TLR9 in many immune cells. In humans, it's much more limited. It's really in the plasmacytoid dendritic cells and B cells. And there's also a difference in the STING activation pathways between mice and humans. But again, just as with RNA, a PBMC assay to measure the products of the IRF pathways and the NF-kappa B pathway and inflammasome pathways, a PBMC assay will pick up activation of any of those pathways if they are occurring under the experimental conditions that you're using. And again, reporter cell lines are of limited value if you're just trying to detect that. Now, when you design an ASO or an siRNA, of course, you're really not intending to activate these receptors. And so I just think, again, it's important to stress these points that ASO and RNA doses are usually way above the doses that are required for TLR activation. At least if you're using, typical gapmers, you may be giving a patient hundreds of milligrams versus an adjuvant dose of a CpG DNA in a vaccine may be just 1 to 3 milligrams. So you're way above the dose at which activation of these TLRs typically occurs. And again, stressing the point that the reason for this is because these oligonucleotides are internalized into endosomes. And so they have an opportunity, they're concentrated, and they can activate those receptors before they go into the cytoplasm and reach the activity at which they can now start to have antisense or RNA effects. And of course, multiple mechanisms may coexist, and you don't necessarily need to determine exactly which of those receptors are activated. But if you're just using unmodified phosphorothioate oligos, which hardly anybody is anymore, it's a good thing, I think, immune and aptamer effects are very likely to occur in vivo. And many of the published reports in the antisense and RNA fields, at least until recent years, but unfortunately still continuing, lack adequate controls to really determine how much those immune effects are contributing to the biologic activities in vivo. One other point I want to make is that TLRs and other innate immune receptors are expressed in quite different patterns. Expression is compartmentalized among different immune cells. Some receptors like STING and RIG-I are more or less universally expressed. TLR7 and 8 which detect RNA, are fairly widely expressed among cells. TLR9 is much more limited in humans to plasmacytoid dendritic cells and B cells. Again, this is another reason to use a PBMC assay where you have all of those cell types present, and or most of them, so that you can detect those activation patterns. What does it look like once you start to modify nucleic acids? So this is looking at just 2'-methyl substitution in phosphorothioate RNA, where the parent sequence here, the 1505 sequence, has a motif, UUGU, which is a very good activator of TLR7 and TLR8. And this is in a human PBMC assay where you're measuring interferon production. And that sequence induces strong interferon response. We're looking here, the highest concentration reached is 2 micromolar. And one point to make is that when you're injecting an oligo subcutaneously, the local concentration of that oligo can be very high. It may be well above 100 micromolar. And typically in these experiments, you're only going up to 2. So again, you have to realize the concentration in vivo may be very high. Anyway, the good news is that whether you make a modification at any position there with the 2'-methyl, it essentially abolishes the activity, at least within this dose range. Modifications outside of that also tend to reduce the stimulation, but by a smaller degree. And so the focus typically is on the U's in modifying RNAs. They may be the most common base involved in stimulating these receptors. And of course, there are published reviews. This is an excellent one from Smith and Zane, summarizing the data with different receptors and the ways that you can avoid activating those receptors. And the good news is a broad range of different modifications does reduce at least the stimulation of those receptors. Now, I say reduce because I'm not sure that they can actually be fully eliminated. And so, as Patrick mentioned, that's one of the reasons why it's so important to bring your dose down using high potency ASO or siRNA designs that don't require as much. So you're less likely to trigger these. Another approach is to use a different backbone that doesn't interact with proteins to anywhere near that degree. The phosphorothioate backbone, of course, having a lot of protein binding, and that's one of the reasons why we like its PK effects. PMOs, on the other hand, this does not look like a nucleic acid to innate immune receptors. They do not interact with PMOs. And so you have a much broader safety margin here before you're going to be activating innate immunity. On the other hand, because these don't activate risk, of course, you can only use them as steric blockers, but it's possible to dose them to a very high level. Now, one of the modifications of PMOs that is trying to build on the safety of the backbone and improve delivery are the peptide conjugated PMOs, where you add a peptide on, a cell-penetrating peptide. Of course, the peptide may introduce toxicities, and that was certainly one of the issues initially when the first PPMOs were taken into primate safety testing. Kidney toxicity was an issue. And one of the other issues that I think will need to be keeping in mind as PPMOs move through the clinic is that they may very well induce the production of antibodies against the PPMO, even if the PMO part of itself does not interact with innate immunity. It'll take time to see how that works, but I certainly think it's an extremely promising and interesting class of drugs for therapeutic development. So formulation, won't say a whole lot about this. It's probably obvious that formulations can alter PK and biodistribution. And just one point to make is that the very same formulation, Smarticles, were used by 3 different companies with 3 different nucleic acids, and they saw very different safety profiles. When a DNA cargo was loaded by Prony into Smarticles, there were mild immune symptoms. When an unmodified RNA that had use and could potentially activate TLR7 and 8 was developed by Myrna, they saw cytokine release syndrome in patients. They had to give high-dose steroid prophylaxis in order to dose people with Smarticles. And then when a modified RNA was used by Mina, those were generally well tolerated. So making the point about the importance of the cargo and thinking about that too. So finally, coming back to in vitro screening, and just to summarize here, the assays that we think are especially useful for screening ASOs or siRNAs are to use human PBMCs at a much higher concentration than is normally used. The plasmacytoid dendritic cells especially seem to like to be crowded, and using a round bottom plate makes them happy. And just 24 to 48 hours at that will be adequate. For whole blood assays, we've tried those too. In our hands, they don't seem to be better. Other people do like them better. You could consider trying them both. If you use whole blood assays, you generally need to go to much higher oligo concentrations in order to get efficacy. The red blood cells may be binding a lot of them. And then in terms of what to screen for, as I mentioned before, I would definitely want to include a type 1 interferon to measure IRF or detect IRF activation. And then for the NF-kappa B and inflammasome pathways, that's generally pretty easy to measure too. Other markers can be included. You can use Luminex or other multiplex assays. You may or may not gain additional information from that. And yeah, that's for in vivo screening, I do want to make a couple of points about in vivo screening of the ASOs. In mice, although interferon alpha is a very sensitive in vitro marker, it really doesn't seem to get into the circulation. And so to detect induction of the IRF pathways, we use an interferon-inducible chemokine. You can use any, but CXCL10 is a very sensitive one. And that is more sensitive in the blood. And then the other point is on the timing of when you look in the blood for this. In some papers, people have looked at 24 hours, and I think that's too late to detect this. Hepatotoxicity and renal screens have already been talked about as well. So just summarizing the key points, phosphorothioate can activate innate immunity at low concentrations, include controls. And good luck with your experiments, and I'll be happy to take questions. I guess we're doing that at the end.